Welcome you all to this Cryo-EM course. Last class we discussed about how you can what kind of grids we should use it for negative staining temp purposes as well as for Cryo-EM also. We also discuss about how we should handle the grids. When you will handle the grids, we should wear gloves or not, and same time how we will pick up all these grids before use it for carbon coating or glow discharge. As well as we also discuss about how we can use these grids for Formovar coating or some kind of plastic coating. I already showed you those things, that what kind of chemicals we can use it for coating the grids. Now next part is the carbon coating of these grids, because carbon coating is also very important on top of that Formovar, because if we have that coating, it will stabilize that grid and electron beam is not going to damage our TEM grids. That is the reason we have to focus on that particular part. Let us take a look what is happening there. This is a carbon coater. I selected this diagram from the company website, where you can see this is a CC7650 carbon coater. What do we have in this carbon coater? We have a particular chamber here. We have a particular chamber here, this glass chamber, and there is a sample stage. In this sample stage, we are going to keep our grids. If you remember properly, in the last class what I showed you, that we have a paper. What do you have there? If we just make a drawing quickly, so we have some kind of grids on the paper. So if we just consider that this blue is our paper, what do we have? So this is our paper, and on this top of this paper we have this circular copper grid. So there are many copper grids are all attached here. So if I just show you that, okay, so these will be the grids, right? Now what we can do, those grids we are placing this one. So we have to take this one when it is completely dry. So we can wait for 1 day or 1.5 day, and then this glass chamber you can keep it under vacuum. So most of the time we have to keep this system under vacuum. There will be some kind of roughing pump, and then fine pump will be connected and keep this chamber at a very high vacuum. Otherwise, when there is a carbon will spread, it will not spread on top of our grid properly. So if there is a lot of air and if there is a motion between this here, it is going to affect the deposition of the carbon. That is the reason this entire chamber will be under vacuum, high vacuum. Now if we want to open this one, what we have to do, we have to first vent the valve, so some air it will go, and this the glass chamber, we can open it completely. We can open this glass chamber completely, and then we can place this one here. So what we can do here, there will be my paper where grids will stay, and now you can use some kind of sellotape kind of thing and stick that, and this will be a flat paper and grids are all flat. So this is very important, this paper and the grid should be flat, so things will spread properly. If this is a uneven, if the paper is not flat like that, if the paper is like this kind of uneven, so this is up, this is up and down, so then the carbon deposition will be very uneven. So this should be very flat, it sticks, otherwise it will just move around here and there. So you have to fix this, and then you have to put this gas chamber back, this complete chamber back, and there is a sealing is here, the gasket is there. So this gasket is going to cover, this is the another gasket is here which is you have to completely tighten both of these things. Now when this is done, you have to keep this one for under vacuum for some time. When this entire chamber will reach certain vacuum, what you can do, here you can see there is a chamber pressure, you can measure that vent. So this valve, it will show you what is the value of your vacuum here. If you reach a high vacuum, what you can do, now you have this voltage, you can see the carbon coated, you can change the voltage, how much electric current is passing through this carbon rod. Inside what will happen, there will be a two carbon rod will be there. One will be thicker carbon rod and another one will be a comparatively thinner carbon rod. So that will touch here and that will pass the electric current and changing the voltage here, and electric current will pass through here to here, because here the thickness is much higher and here thickness is much lower, and there will be a resistance which will create some kind of vibration and it will start spreading the carbon layer, amorphous carbon on top of that grids. So in the later slide this will be clear, because here that carbon rod and all these details are not mentioned. If we just go to the next slide, you can see now here, now we are going to focus only this particular part inside. So here we are showing you the entire system, how this entire system look like and how we are keeping the grids, and now we are going to focus on only the central part. So if we just come here, you can see it here. So as I mentioned that there is a two different carbon rods will be here. This rod will be sharpened carbon rod and this will be unsharpened carbon rod, and both the cases the edge of this rod. So if we just consider that here this will be the thickness of this carbon rod. So this will be the thickness of the other carbon rod. If we just go all the way down and then after sharpen, this will be the two carbon rod. So one will be the thick and then we'll sharpen that, and both the cases that surface of this both the carbon rod will be very shiny. So it should not be very rough. Then what will happen, if it is very rough, when these rods are connect with each other, so if this one is the thin rod, if this is the thin rod and if this is the, so if this surface is very uneven, what will happen due to this friction, because when the electric current will flow from here to here, so in this particular case here your electric current flow is going from this this carbon rod to this thin carbon rod, and there will be a resistance will be very high because the diameter is changing drastically, and that is basically giving some kind of friction between these two rods, which is helping us to generate that amorphous carbon and that will spread. Now if this surface area of this carbon rod and this carbon, basically surface area of this carbon rod and surface area of this carbon rod, if it is uneven, when there is a friction, this rod may break here immediately, and that is the reason we have to use this rod and make it very shiny in both the side, and then we have to attach them in this way that there will be a power supply will go from this end, and here you can see this is the thinner rod and this is the thicker rod, and then it will, when this electric current, it will increase that, when you will change the voltage, if you just remember that last slide, here we are changing the voltage, we are increasing the voltage. After a certain point you can see there is a glow and carbon will start spreading all over, and carbon will deposit on top of this grid. This is how we are coating the carbon on top of that grid. Here this is the same schematic diagram, this is the portion we are showing it here, what should be the thickness, and all the details are mentioned, that this is the sharpened rod, this is the sharpened rod, where this is the thinner carbon and this is the thicker carbon, and it is mentioned that what should be the thickness or radius of that particular rod, and what should be the length of that rod. So here I want to show you one example, because I bought this carbon rod from that laboratory. So if you pay attention to my hand, you can get that details how we can make this. So here you have a 2 carbon rod I am holding. So here you can see the thickness of this one is a much thinner than this one. So how these rods are attached with each other? These rods are attached like that, and there is a spring which is holding this rod even after carbon evaporation. When this particular rod is shorter and shorter, they will be always connected with each other, and due to this vibration, this kind of friction, it will generate some kind of carbon on top of that grid. So this is how generally we are doing carbon coating on top of our grid. So this is little bit a laborious task, that okay, first day you have to coat your glass slide or mica with formable coating or some kind of plastic resin, and then you have to put the grid on top of that, and then you have to keep it for 1 or 2 days. When it will completely dry, you can do the carbon coating. When you are in this particular stage, when this carbon coating is done, we can go for the next, that we can use it for negative staining purposes. So let us take a look actual equipment, how it should look like. So that is very important, how this equipment look like. So here I am showing you a carbon coating equipment. So this is called the Coramtech company. So this is the chamber what we are talking about, the glass chamber. So there are gasket is here, one in the schematic diagram it is showing, one is here and one is here. When you want to open only this particular lid, we can just under vacuum we can open. If we want, we can open this entire glass chamber also. Here you can see this glass chamber is little bit blackish in those areas. When this carbon evaporation is happening, it will coat the grid inside here, but same time there are some carbon it will deposit in the side of this glass chamber, and that is the reason these glass chambers are black. Sometime we have to open this glass chamber and we have to clean it to visualize inside how the coating is going on. So here this is the window where we can set up different kind of parameters. We don't have to change anything manually. Here if we just set up all the parameter, that this is the current we want, this is the voltage we want, this is the pressure, everything will be regulated. Here you can see a pump is connected here, as I mentioned earlier that this chamber will be under vacuum. If it is not under vacuum, your carbon will be not evenly distributed on top of the grid. That is the reason you need a very good vacuum before you do the carbon coating. So let us take a look inside. As I mentioned couple of times, here I already showed you these 2 carbon rods, right. Here you have a carbon rod, that is the diameter is bigger than this particular rod. This is the serpent rod, and there is a spring which is basically holding these 2 rods tightly. So here there is a 2 screw is there, which we can use it to remove this carbon rod or insert this carbon rod. So here this is the thicker, this is the larger diameter rod, and this is the smaller diameter rod. They are connected with each other. Now this is the same view in a different scale. So you can see this entire setup, how it is looking from the top. If you go to the next, here you can see that we just want to show you, this is the rod and this is the spring is holding these 2 rods together. If you pull this rod in that side, they will be separated. So in this particular case, you can see this the serpent rod and the main rod, they are separated from each other. So because this is the spring and I am holding this spring in such a way they are not attached with each other. So now based on that how much I want this length, based on that I can adjust all these screws and they can touch with each other before we start the carbon coating. Now here this is the entire setup again, and here it is showing you how this is connected. So now in this particular case you can see that okay, so this is the inside setup. Then I showed you that okay, well there will be a plastic coating that will be separated. Now when this glass slide is there, so first this is the grid with a plastic coating with the paper, and now we are using this glass slide to hold this paper, so this paper should not move around anywhere. Or if you want, you can use some sellotape also to keep this one on this platform, and then you close this entire setup. You are holding this Formovar coated grids using this glass slide, and then you use carbon evaporation and coat the grid completely. So this is the procedure how you can do the carbon coating. Now the question it is coming that okay, we use the plastic coating and then carbon coating, and we are going to use these grids for our biological science sample preparation. How we are going to use that for our biological science sample preparation part, and if we just use this grid directly and if we add sample, what will happen to that sample? So can you see that sample properly? So the main problem it is going to come that we all have to remember that we have a some kind of plastic coating, and then we have a amorphous carbon, and whenever you will add water or your buffer solution, the carbon is hydrophobic, that plastic coating it is hydrophobic. So then there will be, it will not water or buffer will not spread properly on top of your grid. So it will just making a different kind of curvature. When your grids are hydrophilic, whenever you will add the sample it will spread properly. One thing we also remember, for carbon coated grid there is a grid, there is a two surface, one is matte surface and one is the shiny surface. So most of the time this shiny surface we are using for sample preparation, because that is the area where the carbon coating is done. This shiny surface we can use it, shiny surface at the facing towards us, and then we do the glow discharge and then we use this grid for sample preparation, and all the time we are keeping shiny surface up. For the cryo grid, quantifoil grid, we are just using the opposite, we are not using the shiny surface up. So in this particular case, that what I told, that grid is already coated with plastic and then carbon, both are highly hydrophobic, and whenever we will give the water, water will not spread, and if sample is not spreading on entire grid, that will be really not good for the sample distribution. And also if it will not spread, it will not attach with the grid properly, then what will happen, the excess solution when we will try to remove it, you will see that most of the sample is blotted away. So that is the reason somehow we have to make that grids hydrophilic. That procedure, basically making our grids hydrophilic, it is called the glow discharge procedure. All the time it is better to use our grids and make it hydrophilic. Sometime we need a particular equipment to make that grids hydrophilic. Lot of time we cannot find that particular equipment, we can find that electron microscope in many places, but we may not get the glow discharge unit. If glow discharge unit is not there, your sample will not spread properly, even your staining will not absorb the stain properly, your grid will not absorb the stain properly, and then your stain will be very uneven. So we have to avoid this situation. So if there is a glow discharge units are there, we will do the glow discharge, that I am coming the later. But there are some other procedure, there are many papers published on those things. Some cases, some particular protein we or cell or bacteria, we can clean our grid using the organic solvent also. So, making the grid hydrophilic is a good practice and we need that one. So, it is good for your specimen, good for your imaging and good for the microscopes also. So, generally that organic solvent we can use it, that acetone or ethyl acetate. So, you can sometime you can also use chloroform or C2H4Cl2, that ethylene dichloride also. So, either chloroform or ethylene dichloride or ethyl acetate or acetone. Some cases people, researchers are also using this one, that they are keeping the grid in the petri dish, and most of the biology lab we have the biosafety cabinet. When you have the biosafety cabinet, we have the UV lights in the biosafety cabinet, and try to keep those grids close to that UV light, and if you keep it for 1 hour or sometime 2 hours, based on the strength of that UV light, your grid will be bit hydrophilic and that will serve your purposes also. So, experimentally this is well established that sometimes this UV light is really helping, and you can definitely, if you do not have the glow discharge unit, you can definitely try that UV light in your biosafety cabinet, but remember that it is going to affect the lifetime of your UV light. If you keep it on for 2 hours 3 hours unnecessarily, that is going to decrease the lifetime of your UV lamp. And also you remember that you have to keep the grids very close to the UV light. So, it should not be far away from the UV light. If it is close to the UV light, even if you keep it within 3 or 4 inch within that UV light, the effect will be much higher. Now, let us take a look that glow discharge unit. What is that glow discharge unit? So, here I am showing you 3 different types of glow discharge unit, where you can see some kind of glow, that is the purple color light. This is a plasma. So, glow discharge unit is a glass container. It is attached with the unit. What it is happening, what is happening here, you will place the grid somewhere here on top, somewhere in this place or somewhere here. So, different equipment has a different kind of setup. Now, you have to place this glass chamber properly. So, there will be a gasket here which is tightly packed, and then you have to keep this system under vacuum. When you reach to a particular vacuum, so if I go to the next slide, you can see this is the unit. Basically, this is from the Pelco or TED Peller, where you have, this is the chamber and a pump is attached with the system. When this entire chamber will be under vacuum, and you need a very good vacuum, when you reach a good vacuum, that is the time it will insert little bit normal air, or sometime people are researchers are using nitrogen or sometime argon or some other gases, and then there will be a voltage difference between cathode and anode. When there is a voltage, very high voltage difference, there will be this gas molecule will be ionized because of this cathode and anode, and when they will ionized, they will start spreading all over this gas chamber, within this chamber, and there is a our grids will inside this, and within this chamber this ion will deposit on top of your grid. So, here you can see there is a glow, this is the plasma, and then your grids are sitting here, and ionized gas will spread all over within this gas chamber, and then those ions will deposit on top of your grid, and it will make your grid hydrophilic. And now, from this one, that okay, what will be the voltage potential difference? So, you can get all this information, this electrode, there will be a potential difference, and also initially you need a high vacuum, and then very small amount gas molecule will be inside this chamber. And when you will do this exercise completely, it will make our grid very hydrophilic. When this grid will be hydrophilic, then we can use this grid for our other purposes. So here there is another equipment is also available. So for the plasma cleaning, also this type of equipment we can use it to clean the cryo holder or normal TEM holder also. Now here the plasma treatment, how it is helping us. The plasma treatment is very important because this plasma created by ionization of a low pressure gases in air, glow discharge, it may be oxygen, argon or hydrogen. Ions interact with surface to remove the organic material on top of that grid, whatever we are coating, because we have some organic molecule or plastic coating, and that is going to remove contamination, and contamination will remove completely from the grid and it will make the grids hydrophilic. So these are the way that we can make that grid hydrophilic. So sometime other molecule can be introduced to alter the surface. So amylamine is the another molecule that we can introduce and that can change the surface of the grid. So this is the main reason to use the glow discharge to change the surface property of the grid. So sometime we can use positive glow discharge, negative glow discharge. Based on my protein we have to change this, and lot of times this is also very important parameter when you will do the plasma treatment. How long we are going to do the plasma treatment, what milliamps we are going to use as a current source to ionize the gases, all these things matters. So sometime if you are using the plasma treatment for very long time, it is going to damage your plastic coating and the carbon layer. And sometime if we are using it very low, that is also going to not ionize the grid properly. Sometime if we have a different type of plasma treatment, some protein will go away towards the carbon, it will not stay properly. So those things really matter. This plasma treatment is also very important when you will do the cryo microscopy sample preparation, because that time sample should go and stay inside the hole. If you remember previously I discussed about these things, that what do we have in our cryo grid. So if this is our grid and we have a individual squares, so here we are paying attention to one individual square. What we have? We have this kind of holes, and this is the carbon layer. So here this is the carbon layer and this is literally a hole. This hole size may be 2 micrometer or 1.2 micrometer or 0.6 micrometer. Now when you will add the sample here, what will happen inside that hole, due to surface tension only buffer and sample will stay inside the hole. Now what we are doing a glow discharge, and when we are doing a glow discharge we are trying to change the property of the surface property of the grid. So some kind of ion is deposited here, that is also going to affect our freezing parameter when we will try to freeze our sample. So in some cases this glow discharge, when we are expecting that our sample will stay inside the hole, basically because of these different types of glow discharge, sample will go back to the carbon area. So it is staying, because this is the carbon area outside our hole, right, and sample will move from hole to the carbon area, or it will be completely blotted away, and sometime it will just stick in this area. So that is the reason how long we are going to do the glow discharge, and what when we are ionizing the gas, what kind of milliamps we are using, those things we have to pay attention. Sometimes this negative and positive glow discharge is also helping. Otherwise sometimes some kind of other different material can also make your grid hydrophilic. Based on your project, based on your protein sample or biological science sample, you have to decide and you have to optimize those parameters in the laboratory. So, it is very difficult to give you the exact parameter for your purposes that you have to decide in your lab, and you have to optimize those parameters. Most of the time, we use that 60-second glow discharge for quantifoil grid. For the C flat grid, I am talking about cryo-EM. For the cryo-EM sample preparation, for the C flat grid, generally we are use the much shorter time frame, some 20 seconds glow discharge. For the carbon coated grid, 30-second glow discharge, and then some kind of milliamps we have to use it, and that we have to optimize based on our equipment, based on our project, and based on our grids also. So, now I just want to show you one more important part, that here we can see it that okay, if we do not do glow discharge and if we do the glow discharge, what will happen? If you look at that, when we did not do the glow discharge, the curvature of the sample is different. So, you can see this is the curvature between the grid and your sample. Where we have the glow discharge, you can see the curvature is different. So, this particular angle here and this particular angle is different here. And in this particular case, you can see when we did not do the glow discharge, sample did not spread all over the grid. It is just trying to stay in one particular area. Where when we did the glow discharge, it just spreaded all over. And it is basically distributing the sample in a very uniform way, where if we do not have the glow discharge, it will not distribute the sample properly, and that is the reason I told, if we just blot this kind of grid, most of the sample will go away, and your staining will be very uneven if you do not have the glow discharge. But if you do the glow discharge, your sample will distribute properly, your grid is hydrophilic, so your stain will also distribute properly, and your most of the time your staining will be very even. So, that is one of the major reason we should use glow discharge even if we are making our grids in our home or in our laboratory, or if we purchase those grids from the company. The glow discharge is very important. Either you use equipment or you use a UV light or some chemical treatment, you have to clean your grid, you have to clean the contamination and plastic layer before you add your sample on top of this grid. So, this is all for this class, and in next class we are going to discuss about how to do the negative staining. Thank you all for your attention.